Thank you for joining our webinar today on deep immune profiling to reveal IO biomarkers and understand resistant mechanisms. Today, the main R&D focus in IO are the ones listed here, which are enhance T-cell activity, increase the immunogenicity of tumor, release immune brakes mechanisms, change immune cell polarization, and promote T-cell trafficking into the tumor. But the key word here is combination. Everyone is looking at combination of different treatment modality, both at preclinical and clinical stage. In clinical, actually, based on this review published last year, there are about more than 3,000 clinical trials built on the pillar of PD-1, PD-L1 blockade. As you can see here, there have been a dramatic increase since 2 years. So a lot of resources are used in clinical trial trying to figure out more efficient way to treat patients. So can't we better use preclinical models to address some of those questions? Immune checkpoint inhibitors have revolutionized the cancer therapy landscape. However, adaptive response can be damped by immunosuppressive mechanisms within the tumor, and unfortunately, response to immunotherapy remains restricted to a subpopulation of patients. This is due to the complexity of the immune system and tumor escaping the immune surveillance and our lack of knowledge on refractory mechanism of action. So we need rational strategy to remove barriers to effective anti-tumor response. Building on what we learned from checkpoint blockade, multiple investigational strategies have since aimed to overcome the immunosuppressive pathway that protect tumor from productive anti-tumor immunity. This strategy include direct tumor targeting through antibody like immune checkpoint inhibitors, also using systemic or in situ cancer vaccine, using adaptive cell therapy, activation of immune cells such as T cells, macrophages, or NK cells, and targeting the tumor microenvironment through multiple mechanisms to reduce the number of regulatory, adaptive, and innate cells and block immunosuppressive metabolite and cytokines, and also disrupt the tumor vasculature. At Crohn, we have done extensive characterization of four models in order to help to investigate the mechanism of action of immunomodulatory drugs. So here, this is an efficacy profile of the three main immune checkpoint antibodies, so the PD-1, PD-L1, and CTLA-4, where we have looked at tumor growth inhibition across a range of syngeneic models. And as you can clearly see for either anti-PD-1, PD-L1, or CTLA-4, the response of immune checkpoint in those models could be very variable, with some models showing high response to immune checkpoint inhibitors, whereas other models are very refractory models. In addition to the pharmacology data presented in the previous slide, we have done also extensive immunocharacterization of whole model. So here I presented immunoprofiling data at baseline, so looking at tumor size which are around 100 millimeter cubes, and different immune cell population have been analyzed, looking at T cells population, so CD3, CD8, CD4 marker, and also looking at myeloid compartment and macrophages and NK cells. So we can clearly see, looking at different tumor models here, that immune cell infiltration could be very different across the different models, with some models showing high level of T cell infiltration, whereas other models show very low T cell infiltration. Also, immunosuppressive cell population could be variable, with some models with high Treg, for example, or other model showing high MDFC level. In the study case presented here, we have investigated the mechanism of action of anti-PD-1 in 4 different models. 2 colorectal model, MC38, CT26, and 1 liver model, HEPA-16, and breast cancer model, EMT6. So in this study, we have looked at the efficacy of anti-PD-1, so this is the orange line, compared to the PBS treated group. So this is the anti-PD-1 baseline efficacy. And then we have investigated the impact of immune cell depletion on the efficacy of anti-PD-1. So we have looked at depletion of CD8, CD4, NK cells, macrophages, and Treg. So this is illustrated in this graph here, and this is summarized in the next coming slide. The TGN impact with immune cell depletion compared to anti-PD-1 treatment is illustrated in this graph when we compare the tumor growth inhibition percentage change compared to anti-PD-1. With the baseline of anti-PD-1 treatment compared to the PBS group expressed by those values. So what we can clearly see highlighted in the green boxes for the 3 model MC38, C226, and EMT6 is that when we deplete our CD8 T cells, we completely abolish the efficacy of anti-PD-1 treatment, which is not the case in HEPA-16 model. However, when we deplete CD4 T cells in HEPA-16 model, we do see also here a strong reduction of efficacy of anti-PD-1 treatment, which is not the case for CT26 or MT6, and even in MC38 we do see an increase of the efficacy when we deplete CD4 T cells. In addition, Treg depletion have also some impact in EMT6, CT26, and particularly in MC38, where we do see also increase of the efficacy of anti-PD-1 treatment when we deplete Treg, whereas there is no differences in HEPA-16 whether the Treg are depleted or not. In order to understand better the impact of immune cell depletion on anti-PD-1 efficacy and how the immune cell depletion modulates the tumor microenvironment, we have done some deep immunoprofiling of the tumor following anti-PD-1 treatment. So this is illustrated in the heat maps below that indicates the percentage change relative to anti-PD-1 treatment group, and the red number indicates statistical significant changes. So for each model, in the first column, this is anti-PD-1 after CD4 depletion, second column anti-PD-1 and CD8 depletion, the third column anti-PD-1 and macrophages depletion, and the fourth column anti-PD-1 and NK cell depletion. For MC38, MT6, and CT26, we can clearly see that after CD8 depletion, we do see a significant increase of M2 macrophages, which is not the case for HEPA-16, where we don't see any modulation after CD8 depletion. However, for HEPA-16, after CD4 depletion, we do see a significant increase of the myeloid-derived suppressor cells, which is not observed in the 3 other models. So complementary to the flow cytometry immune profiling data, next generation sequencing based marine targeted gene analysis allow deep immunogenomic profiling of tumor immune interaction that could be used for different purposes. For example, elucidate the drug mechanism of action, develop biomarker signature of response, understand the drug resistance, and inform the next decision in your drug development process. So in order to effectively mount an anti-tumor response, understanding the teaming contribution of the tumor immunity is key, And the evaluation should not only be done only in the clinic, but it needs to be implemented across the range of preclinical models that are studied for immuno-oncology. Current genomic assay, including microarray-based technology, show lower sensitivity compared to next-generation sequencing and are limited in mouse immune cells and lineage that are accurately characterized. A more efficient and robust analysis method is needed to support cancer immunotherapy development. At Crown Bioscience, we have developed a targeted RNA-seq panel of 1,018 genes, specifically looking at genes that are associated with tumor immunity, enabling rapid transcriptomic insight into key immune cell population and IO pathway and process in the team. The 1,080 genes cover immune cell signature that are listed here, including surface marker, transcriptomic immune cell specific biomarkers, and key pathway and the interface of the tumor, the TME, and the immune response. And the functional annotation represented in the panel are demonstrated here as well. So the information are also available as annotated gene list for those that are interested. Coming back to our study case, we have looked at transcriptome analysis of MC38 and EPA16 tumor. The principal component analysis show a large difference for PC1 between MC38 model and EPA16 model. Also, looking at differential gene expression, we can clearly notice here that the number of differential expression genes are high between EPA16 and MC38. The gene set variation analysis is presented in this slide. So for each graph on the left side, the four boxes correspond to EPA16 with respectively the PBS group, anti-PD1, anti-PD1, and CD8 depletion, and anti-PD1 and CD4 depletion. And the four boxes on the right corresponding to the MC38 model. So if we look particularly on interferon-gamma response in MC38, we can notice that anti-PD1 treatment increases the activity of interferon-gamma pathway compared to the PBS group. And CD4 elimination further enhances on the interferon-gamma pathway, whereas this is the completely opposite effect when we deplete CD8 in MC38. In EPA16, the loss of CD4 causes severe reduction of interferon-gamma response to anti-PD1, and those results both in EPA16 and MC38 are consistent with the pharmacodynamics and the TILS pharmacokinetics results that have been presented in the previous slides. In addition to the immunoprofiling and transcriptomic analysis, proteomic analysis was conducted by Biognosys to provide further insight. I will present briefly the result, and that will be detailed by Johan in the next presentation. Unsupervised clustering data are presented in this graph comparing both EPA16 in red here and MC38. What we can clearly notice is there is a large difference between the two different tumor models at proteome level. Also, what we can clearly see here is that group 4 in MC38 is the only group that cluster together. In-depth proteomics analysis are presented in this graph, where we are focused on interferon-gamma pathway, but also protein involved in antigen presentation machinery. So for each graph, the four left boxes are corresponding to the HEPA-16 model, and the boxes on the right corresponding to the MC38 model. And for each model, the first boxes corresponding to the PBS group, to anti-PD-1 treatment, anti-PD-1 plus CD8 depletion, and anti-PD-1 plus CD4 depletion. So globally, what we can notice, particularly in the proteins involving interferon-gamma pathway that are highlighted in red, is that the basal level for HEPA-16 is higher compared to MC38, meaning that HEPA-16 is more hot tumor profile than MC38 compared to the team. Now, if we look more specifically to MC38 and we compare the anti-PD-1 treatment, so the blue boxes compared to the PBS groups, the purple, we do see that we have enhanced expression of MHC class 1 antigen presentation pathway like TAP1, TAP2, TAP BP, but also MHC class 1 expression at H2K1 or H2Q7. And actually, this is further enhanced when we deplete CD4 T cells that improve anti-tumor immunity and lead to tumor death, which is consistent with the mechanism of action in the previous observation. And also we have observed that interferon-gamma protein is greatly upregulated after CD4 depletion compared to anti-PD-1 treatment alone. Now, if we focus on HEPA-16, we do see the opposite effect, where CD4 depletion have a significant impact on the protein implicated in interferon-gamma pathway, where the loss of the CD4 T cells lead to reduced anti-tumor immunity and tumor death, so confirming the role of CD4 T cells in anti-tumor activity. According to the data presented in the last couple of slides, we can clearly see that anti-PD-1 treatment rely on different mechanisms of action in MC38 compared to HEPA-16. With MC38, tumor shows strong CD8 dependence response to anti-PD-1, and where CD4 depletion leads to enhanced response. Whereas in HEPA-16, response to anti-PD-1 is not CD8 dependent, and CD4 depletion lead to resistance, probably inducing lower immunogenicity and impairing interferon-gamma signaling, and also switching the TMA in more immunosuppressive tumor microenvironment. So in summary, this presentation has shown that anti-PD-1 treatment lead to distinct response in different syngeneic models, and that this response is mediated by different immune cell lineage, where we are focused on two different models, HEPA-16 and MC38, that show that anti-PD-1 is respectively for HEPA-16 mediated by CD4 T cells, whereas it's mediated by CD8 T cells in MC38 model. So deep immunocell profiling using different modality that have been presented, flow cytometry, RNA-seq data, and Proteomics data are very complementary to reveal difference in the TME between models, but also could be very helpful to determine any biomarker discovery and also understand better resistance to immune checkpoint inhibitors. I would like to thank you for your attention, and also thanks Biognosys team for this collaborative work, and I will be happy to answer any question after Johan's presentation. Thank you very much.
Hello everyone, my name is Johan Feng, I'm a scientific director at Biognosys. It's a pleasure to be part of this joint webinar. And in the second part of this webinar, I would like to talk about the application of quantitative mass spectrometry-based proteomics on well-established syngeneic mouse models introduced by Ludovic in the first part. First, I would like to give you a brief structure of my presentation. So following an introduction on Biognosys as a company and our optimized workflow for FFPE samples, I would like to focus on the collaborative case study we've performed together with our colleagues from Crown Bio. And there I will further elaborate on how proteomics data from preclinical models can provide additional insights into mechanism of action and potentially drive novel target discovery. So Biognosys is a technology platform company in the area of mass spectrometry-based proteomics with a global headquarters in Zurich, Switzerland. And since our foundation in 2008, we've dedicated our efforts in advancing mass spectrometry-based techniques from upstream sample processing down to data analytics. And our role as a leader in this area has been showcased by over 20 granted international patents and over 1,400 publications applying Biognosys technologies. And on the other hand, we see ourselves also as the enabler of proteomic tools by providing services to the drug discovery community across a wide range of applications. And in the next 15 minutes or so, I will focus on one specific application example in preclinical models for immuno-oncology. And since you've heard an in-depth introduction from Ludovic, I will just very briefly walk you through the experimental design. So we've profiled two different types of syngeneic mouse models, one representing colorectal cancer and the other one representing hepatocellular cancer, the MC38 or the HEPA-1/6 models. And both models have been treated with PD-1 blockade in combination with either anti-CD4 or anti-CD8 antibodies, respectively. And FFPE samples were then collected from these tumor tissues and then subjected to proteomics analysis. And before we go into the proteomics part, FFPE tissue, which stands for formalin-fixed paraffin-embedded, are actually a predominant technique to preserve tissue samples collected from preclinical and clinical studies due to the ease of handling and storage. And another advantage of that is that analytes in these tissues are stable up to many years. This is especially the case for proteins, which have been shown to only have very neglectable degradation up to a few decades. Of course, this advantage comes at a cost. So namely, all the chemical crosslinks have been challenging for downstream sample preparation. And since these are tissue samples, especially in clinical settings, the sample amount is typically very low and very precious and often subjected to multiple types of analysis. So to address this question, we've developed and optimized a workflow from sample to quantitative data that can start with a little material as 1 to 2 slices of 2 micron curves with approximate area of 1 square centimeter. So this is then processed with an integrated protocol, which involves deparaffinization, removal of the crosslinks, and trypsin digestion. The yielded peptide samples are then profiled by our single-shot hyperreaction monitoring method, while protein and peptide quantification as well are performed with Biognosys Spectronaut software. So applying this workflow on the 40 mice from this case study with 5 biological replicates per condition, we could identify in total 9,248 proteins, which are represented by over 140,000 peptides. And this actually leads to approximately 16 peptides per protein. So these peptides do not only serve as an epitope for identification and quantification, but also contain additional information beyond this. And I would like to showcase this with a 3D model of porphyrin, which is a glycoprotein primarily produced by natural killer cells and CD8 plus cells, which was quantified in this experiment. And the quantified proteins visualized here on the 3D structure in red can provide the potential possibility to explore so-called proteoforms, such as isoforms, truncations, post-translational modifications, and many more. Going back to the mouse models, the first question we asked is whether we can validate the molecular phenotypes on the proteome level. And as expected, the pairwise comparison shown on this volcano plot between these two different models underlines the distinct biology observed here. So for instance, if we look into the upregulated protein in the hepatocellular cancer model, we see several pathways from both the tumor and the immune component highly enriched among these proteins. So some of the example you can see here in these blue box. And next, we zoomed into our panel of known markers quantified in this data set for immune activation and killer cell activity, and to see whether we can match the molecular phenotype actually to the mouse phenotype. So at baseline, we can actually see obvious differences between these two models with higher expression level observed over the board for hepatocellular models, leading to the fact that these actually have a much smaller tumor size. And upon anti-PD-1 treatment, what we can see is a significant boost in both immune activation and killer T cell activity in the MC38 models, with all these markers being upregulated significantly, matching actually the fact that shrinkage of the tumor was observed. As you've heard from Ludovic's presentation and talk, an interesting observation here is that the targeted depletion of CD4 plus or CD8 positive cells have led to opposite effects. While depletion of CD8 cells diminishes the effect of the anti-PD-1 treatment, removal of CD4 plus leads to further shrinkage actually of the tumor. And again, by zooming into a panel of known proteins, including LAG3 and IDO1, which are known proven targets for checkpoint inhibitor in combination therapy, we see a very good correlation between these protein markers and the phenotypic readout that we observe. So with the answers to our first question in mind, knowing that proteomic signatures fully recapitulate the phenotypes, we were wondering how we could leverage actually the expression data of over 9,000 protein in this case to identify, for example, known targets or to find monitoring biomarkers in this particular treatment scenario. So I will only present one option here. Of course, there are different ways in looking at data, but what we select to do here is to look, to zoom in or to focus on these two time points where we observe the biggest effect of the CD4 positive cells depletion. And what we did is to use a method called POSDA. And in brief, this method allows us to identify discriminative variables based on the signature or based on 9,000 proteins that were quantified in this study, and to find which one actually best drives the separation or can best explain these molecular phenotype. So here I'm displaying the top 25 discriminant candidates, and you can see that very interestingly, this blue trend that you saw on the last slide have been found in many cases, again here, for many of these top 25 candidates. And another interesting observation here is that a majority of proteins that shown this trend actually belong to a list or to a category of genes and proteins regulated by interferon. So this is just one of the options to zoom into the data and to look for novel targets or monitoring biomarkers, and these basically this rich resource of 9,000 protein definitely provide more options to zoom further into the biology. To sum up, I hope this concise case study could showcase the power of combining unbiased proteomic profiling and well-established preclinical model systems. And beside the biological insights from the discovery aspect, we can envision that this rich resource of data can also be potentially mined for example, pharmacodynamic markers or stratification markers in a responder, non-responder scenario. And furthermore, given the nature of mass spectrometry-based assays, we hope this can also facilitate the transferability from a discovery setting further into a clinical environment. With that, I would like to thank my colleagues from both the Biognosys team and the Crown Bioscience team that have contributed significantly to this work. And of course, thank you very much for your attention.